Hello friends. Today, we will understand the law of segregation, which is one of the fundamental principles of genetics. First proposed by Gregor Mendel in the 19th century. It states that during the formation of gametes, the sperm and egg cells, the two copies of each hereditary factor or gene segregate so that offspring acquire one factor from each parent. This means that each parent contributes only one copy of each gene to their offspring, which is selected randomly. The law of segregation is also known as Mendel's first law of inheritance. Now, to understand this law completely, let's break down the law and recall some important concepts. Part 1, during the formation of gametes, sperm and egg cells. Gregor Mendel studied inheritance of traits in pea plants. He proposed a model where pairs of heritable elements, or genes. Genes are specified traits. Genes come in different versions, or alleles. A dominant allele hides a recessive allele and determines the organism's appearance. In sexually reproducing organisms, the genes are carried in two identical copies. A copy was inherited from each parent, in the form of a gamete, the egg or sperm cell. These organisms are known as diploid when they have both copies of the genome, and haploid when they are gametes and have only one copy. Though Gregor Mendel was not clear on exactly how the process took place, modern microscopes and molecular techniques have revealed that alleles are separated during the process of meiosis. Second part of law is: the two copies of each gene segregate so that offspring acquire one factor from each parent. Now, to understand this, let's have a look what actually happens in meiosis. Meiosis occurs in specialized cells known as gametocytes, which form haploid cells from diploid cells. To start the process, all of the DNA in a cell is duplicated. This creates two copies of each allele. In this cell, there are now 4 alleles for each gene, although 2 of them are simply identical copies of the original. During metaphase 1 of meiosis 1, these bonded homologous pairs are aligned in the middle of the cell and separated. In doing this, the different alleles for each gene are effectively separated. During meiosis 2, the copies of the alleles will be separated into individual gametes. This ensures that each allele makes it to a new gamete, giving it an essentially equal chance of finding a gamete to fuse with and create a new organism. Due to the law of segregation, each allele is its own entity and always has an equal chance of being passed on to the next generation. I hope that you enjoyed the video. Please subscribe to get videos like this.